After dark, Australian habitats can reveal points of blue-green light. Some animals make light themselves. Others reveal hidden color only under UV illumination. These two effects look similar, but they are not the same. Bioluminescence is light made by a chemical reaction inside an organism. Glowworms, fireflies, lantern sharks, and pyrosomes are true bioluminescent animals. They do not need a torch to produce their glow. Fluorescence is different. Pigments absorb higher energy light, often UV, then release some of that energy as visible color. Switch off the UV source and the fluorescence disappears. Scorpions and many mammals belong in this second group. At Springbrook National Park in southeast Queensland, damp rock overhangs can resemble a starry sky. The lights come from larvae of fungus gnats in the genus Arachnocampa, Australia's famous glowworms. The larva produces blue-green light in a specialized organ near the end of its body. That glow is a lure. Tiny flying insects move toward it in the darkness and encounter hanging silk threads. Each thread is dotted with sticky droplets, like a beaded fishing line. Once prey is trapped, the glowworm hauls the line up and feeds. The display is beautiful, but its purpose is practical, finding food. The glow is strongest when larvae are hungry and conditions are humid. A bright torch or camera flash can interrupt their feeding behavior, so park guidance asks visitors to keep lights low and avoid flash photography. Australia is also home to a glowing beetle called the Blue Mountains firefly, Attophella lignus. It occurs from the Sydney region into southeast Queensland, and its light organ sits in the abdomen. Adults flash to find mates, turning a patch of forest into a brief conversation of light. The flashes are species-specific signals. They are not fluorescence. The beetle's body drives the chemical reaction. Even in ordinary light, the lantern-like area is small and easy to miss. This photograph shows an adult Attophella lignus. The highlighted rear abdomen is where the signal is produced. A related Queensland species, Attophella atra, can look pale and inconspicuous in ordinary light. Australian firefly species differ in form and flash pattern, but they share the same basic trick, transforming chemical energy into cool, visible light. Far below the sunlit surface, Merla's lantern shark occurs off eastern Australia and elsewhere in the western Pacific. It is a small deepwater shark, recorded from roughly 240 to 700 meters. This museum photograph shows preserved Merla's lantern sharks. In the ocean, tiny light-producing organs called photophores cover parts of the body. A preserved specimen cannot display the animal's natural light, but it confirms the real form and scale. In lantern sharks generally, a dim underside glow can match light filtering from above, reducing the silhouette seen by predators below. Researchers also investigate roles in recognition and signaling. Those functions vary by species and should not be assumed from a photograph alone. In Australian waters, another source of light drifts with the current. Pyrosomes are not single animals. Each hollow tube is a colony built from hundreds or thousands of tiny filter-feeding individuals called zooids. Every zooid can respond to light, and the response can spread across the colony. The result is a traveling pulse, as if a soft blue-green wave is moving through a transparent sleeve. Pyrosoma atlanticum has been recorded in Australian waters. Colonies can be small or grow into long cylinders. In daylight, they may look gelatinous and unremarkable. In darkness, their coordinated light becomes the defining feature. The name pyrosome means fire body. Sailors once described brilliant colonies at sea, but today photographs let us separate the animal's shape from the glow. This is a real colonial tunicate, not a computer-generated effect. On land, scorpions create a different surprise. Under ordinary light, an Australian black rock scorpion is brown and well camouflaged. Under UV, compounds in the hard outer cuticle absorb the invisible radiation and re-emit visible blue-green light. This side-by-side UV photograph uses a European scorpion only to demonstrate the visual effect. The Australian species is shown separately in ordinary light. That label matters. It prevents a representative image from being mistaken for the exact animal. All scorpions are known to fluoresce, although the biological role remains debated. The effect may relate to light detection or behavior, but it is not bioluminescence. When the UV lamp is removed, the visible glow stops. The biggest recent surprise came from mammals. In a 2023 study led by Western Australian Museum researchers, every one of 125 mammal species examined showed some fluorescence under UV. The sample covered 27 mammal orders and 79 families. Researchers examined fresh, frozen, and preserved specimens, then checked the emitted light with spectroscopy. Preservation could change intensity, so the study treated that limitation seriously. The platypus was already famous for glowing blue-green under UV. Here, a daylight photograph is paired with a study image. The comparison is genuine, but it does not mean a platypus shines visibly beside a river at night. The greater bilby's pale fur also fluoresced strongly in the study photographs. Pale or unpigmented fur, whiskers, claws, and exposed skin often produced the brightest response across the mammals that were examined. Common wombats showed fluorescent regions too. These study images were made under controlled illumination, which is why their colors look more intense than a casual nighttime view would. UV exposure and camera settings both affect the result. This quenda, or western brown bandicoot, looks quietly brown in daylight. Under UV, lighter areas can return visible color. The changing photograph here is a matched species comparison, not the same collage repeated. The southern marsupial mole adds a striking example. Its pale coat and sparsely pigmented skin fluoresce strongly. The daylight image is a real museum specimen. The UV image comes from the peer-reviewed study. The study also included monotremes such as echidnas, alongside marsupials, bats, rodents, and many other groups. Fluorescence was most common and most intense in nocturnal, terrestrial, tree-dwelling, and burrowing mammals. Why do mammals fluoresce? There is no single confirmed answer. In some species, it may be incidental, an unavoidable property of keratin, fur, or other tissues. In others, camouflage, signaling, or UV protection remain ideas to test. So Australia's animals offer two kinds of hidden light. Glowworms, fireflies, lantern sharks, and pyrosomes make photons through chemistry. Scorpions and mammals transform incoming UV into visible color. Looking carefully and labeling the mechanism keeps the wonder accurate.